Hello, learners! How are you today? I hope you're doing great. By the way, I am Jonamie Altaque, and I'm here in front of you to discuss about tricarboxylic acid cycle. What is tricarboxylic acid cycle? The Krebs cycle, also known as the citric acid cycle or TCA cycle, is a series of reactions that takes place in the mitochondria, resulting in oxidation of acetyl CoA to release carbon dioxide and hydrogen atoms that later lead to the formation of water. This cycle is termed the citric acid cycle as the first metabolic intermediate formed in the cycle is citric acid. This cycle is also termed tricarboxylic acid cycle or TCA because it was then not certain whether citric acid or some other tricarboxylic acid or isocitric acid was the first product of the cycle. However, now it has been known that the first product is indeed citric acid, and thus the use of this name has since been discouraged. This cycle only occurs under aerobic conditions as energy-rich molecules like NAD+ and FAD can only be retrieved from the reduced form once they transfer electrons to molecular oxygen. The citric acid cycle is the final common pathway for the oxidation of the biomolecules, such as proteins, fatty acids, carbohydrates. Molecules from the other cycles and pathways enter this cycle through acetyl CoA. The citric acid cycle is a cyclic sequence of reactions formed of 8 enzyme-mediated reactions. This cycle is also particularly important as it provides electrons or high-energy molecules to the electrons transport chain for the production of ATPs and water. Pyruvate formed at the end of glycolysis is first oxidized into acetyl CoA, which then enters the citric acid cycle. Now let's move on. Krebs cycle location. Where it is. The citric acid cycle in eukaryotes takes place in the mitochondria, while in prokaryotes it takes place in the cytoplasm. The pyruvate formed in the cytoplasm or from glycolysis is brought into the mitochondria where further reactions take place. The different enzymes, we have the different enzymes involved in the citric acid cycle are located either in the inner membrane or in the matrix space of the mitochondria. We have heard the reactions of the cycle. In the TCA cycle, oxaloacetate is first condensed with an acetyl group from acetyl coenzyme or CoA and then is regenerated as the cycle is completed. We have here in the figure 9.1. Therefore, the entry of the acetyl CoA into one round of the TCA cycle does not lead to the net production or consumption of intermediates. Take note, 2 carbons entering the cycle as acetyl CoA are balanced by 2 CO2, exciting. Oxidative decarboxylation of pyruvate. The major source of acetyl CoA, the 2-carbon substrate for the TCA cycle, is the oxidative decarboxylation of pyruvate. Pyruvate, the end product of aerobic glycolysis, must be transported from the cytosol into the mitochondrion. This is accomplished by a specific transporter that facilitates movement of pyruvate across the inner mitochondrial membrane. Once in the mitochondrial matrix, pyruvate is converted to acetyl CoA by the pyruvate dehydrogenase complex or PDH complex, which is a multi-enzyme complex. Take note, strictly speaking, the PDH complex is not part of the TCA cycle, but it supplies substrate for the cycle. First component enzymes. The PDH complex is a protein aggregate of multiple copies of 3 enzymes, pyruvate. First, sometimes called pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. Number 3, each catalyzes a part of the overall reactions. See figure 9.2. Their physical association links the reactions in proper sequence without the release of intermediates. Number 2, coenzymes. The PDH complex contains 5 coenzymes that act as a carriers or oxidants for the intermediates of the reactions shown in figure 9.2. Requires thiamine pyrophosphate or TPP, requires lipoic acid and CoA, and requires flavin adenine dinucleotide or FAD and nicotinamide adenine dinucleotide NAD+. Note, TPP, lipoic acid, and FAD are tightly bound to the enzymes and functions as coenzymes prosthetic groups. We have here also the regulation of the pyruvate dehydrogenase complex. Covalent modifications by the 2 regulatory enzymes that are part of the complex alternately activate and inactivate. Example number 1. The cyclic AMP independent PDH kinase phosphorylates and thereby inactivates. Example 1. Whereas PDH phosphates dephosphorylates and activates. See figure 9.3. The kinase itself is allosterically activated by ATP acetyl CoA and in ADH. Therefore, in the presence of these high energy signals, the PDH complex is turned off. Note, it is actually the rise in the ATP or ADP, NADH, NAD+, or acetyl CoA, CoA ratios that affects enzymic activity. Pyruvate is a potent inhibitor of PDH kinase. Pyruvate dehydrogenase complex deficiency, a deficiency in the activity of the subunit of the dimeric component of the PDH complex, although rare, is the most common biochemical cause of congenital lactic acidosis. This enzyme's deficiency results in an inability to convert pyruvate to acetyl CoA, causing pyruvate to be shunted to lactate via lactate dehydrogenase. We have number 5, mechanism of arsenic poisoning. As previously described, pentavalent arsenic or arsenate can interfere with glycolysis at the glyceryl dihydrophosphate. thereby decreasing ATP productions. Arsenic poisoning is, however, due primarily to inhibition of enzymes that require lipoic acid as a coenzyme, including A2 of the PDH complex. Acetoglutarate dehydrogenase see below pictures, and branch chain aceto acid dehydrogenase B synthesis of citrate from acetyl coenzyme A and oxaloacetate. The condensation of acetyl CoA to form citrate. Tricarboxylic acid is catalyzed by citrate synthesis. We have here the figure 9.4. This aldol condensation has an equilibrium far in the direction of citrate synthesis. In humans, citrate synthesis is not an allosteric enzyme. It is inhibited by its product, citrate. Substrate availability is another means of regulation for citrate synthesis. The binding of OAA causes a conformational change in the enzyme that generates a binding site for acetyl CoA. Note, citrate, in addition to being an intermediate in the TCA cycle, provides a source of acetyl CoA for the cytosolic synthesis of fatty acids. Citrate also inhibits phosphoctokinase 1, PFK1, the rate-limiting enzyme of glycolysis, and activates acetyl CoA carboxylase, or the rate-limiting enzyme of fatty acid synthesis. Letter C. Isomerization of citrate. Citrate is isomerized to isocitrate by aconitase or aconitate hydratase. An Fe-S protein, see figure 9.4. Note, aconitase is inhibited by fluoroacetate, a plant toxin that is used as a pesticide. Fluoroacetate is converted to fluoroacetyl CoA, which condenses with OAA to form fluorocitrate, potent inhibitor of aconitase, resulting in citrate accumulation. Oxidative decarboxylation of isocitrate. Isocitrate dehydrogenase catalyzes the irreversible oxidative decarboxylation of isocitrate, yielding the first of 3 NADH molecules produced by the cycle and the first release of CO2. See figure 9.4. This is one of the rate-limiting steps of the TCA cycle. The enzyme is all allosterically activated by ADP, a low energy signal, and Ca2+, and is inhibited by ATP and NADH, levels of which are elevated when the cell has abundant energy stores. E. Oxidative decarboxylation of acetoglutarate. The conversion of acetoglutarate to succinyl CoA is catalyzed by the acetoglutarate dehydrogenase complex, a protein aggregate of multiple copies of 3 enzymes. We have the figure 9.5. The mechanism of this oxidative decarboxylation is very similar to that used for the conversion of pyruvate to acetyl CoA by the PDH complex. The reaction releases the second CO2 and produces the second NADH of the cycle. The coenzymes required are TPP, lipoic acid, FAD, NAD+, and CoA. Cleavage of succinyl coenzyme A. Succinate thiokinase, also called succinyl CoA synthetase, named for the reverse reaction, cleaves the high-energy thioester bond of succinyl CoA. See figure 9.5. This reaction is coupled to phosphorylation of guanosine diphosphate or GDP to guanosine triphosphate GTP. GTP and ATP are energetically interconvertible by the nucleoside diphosphate kinase reaction. GTP plus ADP to GDP plus to ATP. The generation of GTP by succinate thiokinase is another example of substrate level phosphorylation. Note: succinyl CoA is also produced from propionyl CoA derived from the metabolism of fatty acids with an odd number of carbon atoms, and from the metabolism of several amino acids. Next is the oxidation of succinate. Succinate is oxidized to fumarate by succinate dehydrogenase as FAD, or its coenzyme is reduced to FADH2, is embedded in the inner mitochondrial membrane. As such, it functions as complex 2 of the electron transport chain. Note: FAD rather than NAD+ is the electron acceptor because the reducing power of succinate is not sufficient to reduce NAD+. Next, hydration of fumarate. Fumarate is hydrated to malate in a freely reversible reaction catalyzed by fumarase or fumarate hydratase. See figure 9.5. Note: fumarate is also produced by the urea cycle, in purine synthesis and during catabolism of the amino acids phenylalanine and tyrosine. Oxidation of malate. Malate is oxidized to oxaloacetate by malate dehydrogenase. See figure 9.6. This reaction produced the third and final NADH of the cycle. The standard free energy change, or GO, of the reaction is positive, but the reaction is driven in the direction of OAA by the highly exergonic citrate synthesis reaction. Note: OAA is also produced by the transamination of the amino acid aspartic acid. We have here the energy produced by the cycle. 2 carbon atoms enter the cycle as acetyl CoA and leave as CO2. The cycle does not involve net consumption or production of OAA or of any other intermediate. 4 pairs of electrons are transferred during one turn of the cycle. 3 pairs of electrons, reducing 3 NAD+ to NADH, and 1 pair, reducing FAD to FADH2. Oxidation of 1 NADH by the electron transport chain leads to formation of approximately 3 ATP, whereas oxidation of FADH2 yields approximately 2 ATP. The total yield of ATP from the oxidation of 1 acetyl CoA is shown in figure 9.7. Summarizes the reactions of the TCA cycle. We have here also the regulation of the cycle. In contrast to glycolysis, which is regulated primarily by PFK-1, The TCA cycle is controlled by the regulation of several enzymes. See figure 9.8. The most important of these regulated enzymes are those that catalyzes reactions with highly negative GO. Citrate synthase, isocitrate dehydrogenase, and acetoglutarate dehydrogenase complex, reducing equivalents needed for oxidative phosphorylation, are generated by the PDH complex and the TCA cycle. And both processes are upregulated in response to a decrease in the ratio of ATP to ADP.
Thank you for watching.